Welcome to our station. We are thrilled to present you our multimodal decision support system for lung cancer in just a minute. Thank you for visiting our presentation. I'm Sandy Chen. I'm professor of radiology and the director of a research center of AI in medicine, Taipei Medical University. Here is my pleasure to present you our exciting breakthrough technology in lung cancer decision support system. This diagram introduces the ecosystem of precision health development among government, university, and AI industry. Our AI lab, the DeepRay AI, is from the Taipei Medical University. We have government funding support and strong collaboration with AI industry. We have three big affiliate hospitals providing huge clinical data. The government not only support funding, but also computing power by aligning the supercomputer from National Applied Research Laboratory. We'll talk about this in just a minute. Our project has strong collaboration with local startup as also like Ether AI, Finding Company, and EBN Company. Our main theme is lung cancer. That is top killer cancer in Taiwan. As you can see here, the incidence of lung cancer is increasing in both gender. More importantly, the 5-year survival is only 15% because 70% of patients were late stage at diagnosis. Furthermore, there are complex driver genes behind the cancers that precision medicine can play a big role. Lung cancer target therapy is a classical case of precision medicine, where the treatment is according to the specific driver gene mutation. For EGFR mutation, as instance, which comprise 40 to 45% cases in Asia, more common in younger women and non-smoker. The target on tyrosine kinase domains such as exon 19 deletion and exon 21 point mutation, specific tyrosine kinase inhibitor can effectively control the disease progression under certain circumstances, such as a patient with cancer with or without the brain metastasis. It is important for the late stage patient because surgery often is not an option and the survival time is short. And the genomic analysis require even more time for a drug selection. So here is the role emerging for our AI algorithm. So our product goal is to facilitate clinical decision support system, shared decision support in lung cancer, and assist the physician in detection, classification of a lung cancer, thereby help predict a drug response and prognosis. We have 6 different core for this AI support system. Here we present only imaging core, including the CT audio, autoradiogenomic, and pathology pathogenomic. So here is the primary design of the decision support system. There are 6 core where each data set input in any of the core model can produce decision support prediction. You can see 2 are imaging core we are going to discuss in more detail. The other is such as a demographic module and the genomic module and the clinical trial matching module. Finally is the drug response prediction model. The functionality of the module are based on actual clinical flow by patient's visit. So for example, the first patient visit with data is the diagnosis CT and the clinical data. After tissue biopsy, the second visit is added with pathological data. So follow this flow, the decision support system can offer advanced prediction for patients' earliest possible treatment choice. such as target drug selection. The development of our CT radiogenomic and Pasogenomic modules are based on thousands of expert-labeled imaging data plus semantic labeling by incorporating the most relevant quantitative and descriptive item into the training. Thus, an automatic text report can be generated through natural language process technology. Here I summarize the developmental process. The lung cancer data were provided by our 3 affiliate hospitals, and the CT and pathological imaging were labeled by board-certified radiologists and pathologists. Then the AI algorithm were directed to produce an auto report. This is our CT module for nodular detection and classification. We called it lung rate. Lung rate featured auto segmentation of a whole set of DICOM CT imaging, about 300 slices. The whole process is fully automatic. As you can see here, this is original and this is after the algorithm. The nodule can be auto-detect. After auto-detection, the next step is to achieve auto-classification of the detected nodule by their radiomic features. Our algorithm, as compared to the other several machine learning methods, can reach about 98% accuracy in the classification, the top row. So literally speaking, our lung rate model can do auto-detection, the auto-classification, and auto-reporting in just one stop. So here show you how lung rate platform can do in real-world case. First, we pick up one case and their CT DICOM data. Then a couple of seconds, the system will tell there are 2 nodules detected and their individual characteristics such as the size and the lung rate gradient. Lung rate gradient is the international management system that advise the physician the best management for nodule. For example, if the nodule need to be biopsy or to be followed. Here is a quick view of the result. The nodule here, the malignant probability is about 98%. The lung rate gradient system is 4A, means that a further diagnostic test, such as needle biopsy, is required. While another nodule is grade 2, thus followed by CT annually will suffice. Now we move the diagnostic lung rate to decision support, the CT autoradiogenomic prototype. This prototype can automatically detect and classify the nodule and infer the possible driver gene mutation by radiomic feature. For this example, this is EGFR prediction module. Our second breakthrough technology is Pasogenomic. This technology enable auto segmentation of the whole glass pathology image as shown here. The lesion slide using a ResNet and auto annotation free imaging training. The technology has been just published in Nature Communication. We use 7,003 pathology slides for training. The model, when tested, showing 94% AUC for segmentation and the classification of adenocarcinoma from squamous cell carcinoma. Here, just show you how the visual idea of whole glass imaging and auto detection of cancer in a large field of the pathological slide, just like a Google map. So the prototype for whole slide EGFR mutation model is illustrated here. You can see when pick up a real-world case, you can see the distribution of the yellow color carcinoma with EGFR mutation. These suggest that the patient can be the candidate for tyrosine kinase inhibitor treatment. So this is a whole new story for this patient. So here quickly show you the summary of CT radiogenomic, the high AUC by using the genetic analysis. The deep neural network EGFR currently has AUC only 0.72, but will be much improved by adding more cases for training. So this is our complete multi-module clinical decision support system. After input the data, the system can analyze the CT, pathology, demographic, and other data, such as the clinical trial along the way. Here is the CT module. As you can see, a lot of functional presentation by input a different layer of the data and some prediction for the diagnosis. Here is the pathological mode. And the other module that I have not mentioned in today's presentation also include the genomic analysis and the clinical trial match, in which case that when the patient had the late stage without any treatment option, the clinical trial like this screen show you that you can find the currently available trial in any hospital in Taiwan or around the world. So our illustration is providing you an idea about how to translate imaging into actionable decision and the invisible visible. Thank you very much for the kind attention.